Alright, so in this video I'm going to show you how to find protein domains using the Spectral DOM software, which I'll be running from the VMD2 interface. So I'm going to Plugins, Structural Bioinformatics Library, Spectral Domain Finder, and I get the window. So let me load the structure. I'll be using one n0u.pdb, and in fact load. So there it goes. Here is the structure. I'm going to display it in cartoon mode here. Alright, and as you can see, this is a big protein. It's actually involving 842 amino acids, and so this is an elongation factor, namely a protein which is involved in the polypeptide chain synthesis at the ribosome level. And so we aim at finding domain. So what is the protein domain? It is essentially a compact region which in general falls on its own and accomplishes one or several functions. And so here it's tempting to believe that we have one domain in this horizontal section, and then possibly one in the middle, one or two in the middle, and possibly one or two above in this upper part. So to find domain, so I have to specify the chain. So this is chain A. I also tell the program I want to inspect between 2 and 10 domains, possibly, and then there are some other options I'm not discussing here. And so with this I'm going to run the calculation, which is going to take a couple of seconds, and we are getting two pieces of information. So in the graphics engine we get a decomposition with color domain. So this is the proposed decomposition, and on the right hand side here in this GUI window we get a curve which is a score curve, and so the higher the better. As you can see, the global maximum of the curve is obtained for k is equal to 6. Therefore, the proposed decomposition is involving 6 domains, and this is indeed what we see in the graphical engine. So one small orange domain here, and then one red, one dark gray, one light gray, one yellow, and one green. So in fact, the algorithm which is used to find domain is a version of spectral clustering. So to explain what's happening here, let me give a metaphor. It's about a community detection algorithm. If you think about a social network with many people interconnected, and these people having few connections outside their own group. So in fact we want to do the same here in the protein to find these clusters, except that the individuals are now amino acids, and the connections of course are made either by covalent interaction, covalent bonds, or non-covalent interaction between the amino acids which are sitting nearby in 3 dimensions. So what we were suspecting was indeed correct, namely one or two, in fact 3 domains in this bottom section, 2 in the middle, 1 above, and as you can see, of course, in between 2 domains we have a sort of gap, and it's the reason why in fact the domain is a compact region. This is what the algorithm has been able to figure out. So if we come back to the curve, so I told you that the maximum is at k is equal to 6, and we have a box plot also for each value of k. The reason for the box plot is that for a given k, the decomposition is run several times because we are internally using k-means, and so we run it several times with different seeds, and we retain the best score, which again here is obtained for k is equal to 6. But of course, it turns out that the best score for k is equal to 5 is in fact almost the same, and so we can from the interface now take a look at the partition obtained for k is equal to 5, and so I put 5, update result, and now if we inspect the result, what has happened, it turns out that the small orange domain which we had in this bottom section disappeared, and so in the bottom section we in fact have 2 domains and not 3. And now, as you can see, this is quite a natural decomposition into 5 domains. We can of course proceed and now take k is equal to 4, update results, and now in fact we still have 2 domains below, but in fact the 2 domains in the middle have been merged. And of course we can proceed, inspect k is equal to 3, k is equal to 3, so now we have a single domain in the horizontal section, and then 1 in the middle, 1 above, and for k is equal to 2, not surprisingly we are getting this bottom section, horizontal section, and this vertical section. So this is what the algorithm does in this diffusion map mode, which again is finding community using the spectral decomposition algorithm. There is a different mode which is called the multiple sequence alignment mode, where one is not using a single structure but several structures, typically two conformations or several conformations of the proteins of the same protein undergoing conformational changes, and the atomic displacements observed between these decompositions are used to drive the decomposition. So I refer you to the paper in order to take a look to this. And so to conclude, we can also go into this directory, which has been preset to host the results. And so if I go there, we are going to find all the files which have been computed. So we get, for example, the score files, which is used to display the quality score plot. We also get VMD files, namely those which are displayed in VMD's graphics engine. And we also get a log file and a number of other outputs which can be inspected and exploited for statistics by scripting, for example, using Python.